In this video, we are going to look at the histological slides of various specimen under nervous system, a total of 7 different specimen. Let's begin with the first one. Okay, this right here is the transverse section of peripheral nerve. How do I know that? All these dots or rounded structures I see right here are the transverse sections of individual neurons. In between these neurons, the empty space is filled up by connective tissue called endoneurium. These neurons, they are present in a bundle, and this bundle is called fascicle. The fascicle itself is also covered by a connective tissue, and this layer is called perineurium. Perineurium is further covered by external epineurium from outside, and in between adjacent perineurium there's internal epineurium. So one last time, I can see neurons arranged in a bundle called fascicle. The fascicle is covered by perineurium. Outside perineurium there's external epineurium and internal epineurium. Okay, in this particular slide, I can see much more number of fascicles. So you can see fascicle covered by perineurium, fascicle covered by perineurium, fascicle covered by perineurium, fascicle covered by perineurium, and perineurium covered by external epineurium and internal epineurium. Okay, so here's the next slide. This slide is that of your cerebrum. The cerebrum contains two parts, the external cerebral cortex and the internal cerebral medulla. The cerebral cortex is made up of gray matter, while the cerebral medulla is made up of white matter. The cerebral cortex is divided into 1, 2, 3, 4, 5, 6. 6 different layers, or the gray matter is divided into 6 different layers. The whole of cerebrum from outside is covered by a meninges, one of the meninges called pia mater, which is also visible under the slide. The outermost layer is your molecular layer, which contains a lot of fibers but very less number of neurons. You can compare the molecular layer with the rest of the layers. You can see that in rest of the layers there are much more dots. These dots, they represent neurons. However, here there are relatively less number of dots. That means there are less neurons but more fibers. So that's your molecular layer. Before I tell you rest of the layers, I need you to understand two different types of cells. One is your pyramidal cell and the other is your stellate or granular cells. Okay, look carefully. Within this histological slide, you can see few triangular shaped cells, triangular shaped large cells. These are your pyramidal cells, triangular shaped, triangular shaped, triangular shaped, triangular shaped, triangular shaped. These are pyramidal cells. The rest dots that you see, these are your stellate cells. Since they are visible as granules under your microscope, they are also called granular cells. Okay, so out of the remaining 5 layers, the external granular layer and the internal granular layer, it's made up of granular or stellate cells, and the external pyramidal layer and the internal pyramidal layer, they are made up of the pyramidal cells. The polymorphic layer is made up of the mixture of both stellate and pyramidal cells. This is the histological slide of human cerebellum. Like cerebrum, cerebellum is also covered from outside by a layer of meninges called pia mater. Inside, like cerebrum, it is also divided into cerebellar cortex, which is made up of gray matter, and cerebellar medulla, which is made up of white matter. The cerebellar cortex contains 3 layers. In cerebral cortex, there were altogether 6 layers. In cerebellar cortex, there are altogether 3 layers. The outermost molecular layer made up of stellate cells and basket cells. The middle Purkinje layer made up of Purkinje cells. And the innermost granular layer made up of granular cells. Now, as compared to the rest of the histological slides of nervous system, cerebellum is much more easier to identify due to well demarcation understanding. So you can easily see these stripes, and based on these stripes you can say that okay, this is the histological slide of cerebellum. look for these stripes change in color with stripes. Okay, next up you have spinal cord. This is really easy to identify. All you have to look for is this butterfly shaped gray matter. So there's gray matter in the middle which makes a butterfly shape, and then white matter surrounding the gray matter. In the middle there's central canal, then there's posterior median sulcus and anterior median fissure. This right here is the histological slide of cochlea. How do I know that? I know that due to the presence of these empty spaces. These empty spaces are nothing but the channels present within cochlea. And there are three channels present within cochlea. What are they? See, here I have the same slide but at a higher resolution. These are the channels. These channels are present in a group of three. Let's take one group. So here's what it looked like. So there's 1, 2, and 3 channels present in a group. Let's see those channels. See, the cochlea, it is highly coiled. When you uncoil this cochlea, you'll see that it contains 3 different channels. 1, 2, and 3. When you take a transverse section of these channels, okay, let me take a fresh page. Okay, so here's the highly coiled cochlea. I straighten this cochlea and get these 3 channels. 1, 2, 3. Here's helicotrema, by the way. When I take a transverse section of these 3 channels from front, this is what it looks like. Channel 1, channel 2, channel 3 present in a group. So that's exactly what you can see under the microscope. You can see channel 1, channel 2, and channel 3. So what are the channel? There's your scala vestibuli, scala media, and scala tympani. These 3 channels are present due to division by 2 membranes called Reissner's membrane and basilar membrane, which are a part of membranous labyrinth. Okay, so basilar membrane, on top of basilar membrane, there's a special structure. On top of basilar membrane, there's a special structure. On top of basilar membrane, there's a special structure, and this is called organ of Corti. It contains tentorial membrane as well as hair cells that are your sensory structures. They perceive the sensation of hearing. So I have the same slide here, but then with this blue spirally coiled arrow. What does it show? It shows the direction in which the cochlea is coiled. At the end, there's helicotrema. This is the slide of cornea. The cornea posteriorly contains the anterior chamber of the eye, which is filled with aqueous humor. From anterior to posterior, there are altogether 5 layers arranged in the order of A, B, C, D, and E. What are these 5 layers? So there's your anterior corneal epithelium made up of non-keratinized stratified squamous epithelium. There's your Bowman's layer. There's a thick corneal stroma made up of type 1 collagen. There's your decimates membrane, which is the true basement membrane. And then there's your endothelium or the posterior endothelial lining. Again, what are the layers? A4, anterior corneal epithelium made up of non-keratinized stratified squamous epithelium. B4, Bowman's membrane. C4, corneal stroma made up of type 1 collagen. Since there are collagen, there are obviously going to be fibroblast as well. D4, decimates membrane, which is the true basement membrane. And E4, endothelium, which is the posterior layer of cuboidal epithelium. This is the slide of your retina. So we saw cornea, which is present at the front of the eye. The sclera, it continues as your transparent non-vascular bulge called cornea. Now there's your retina, which contains photosensitive receptors. These are the cell bodies of the neurons or the cells present in the retina, present in the form of a strip. So these are multiple strips of cell bodies. Let's look at the same retina but at a much higher resolution. So at a higher resolution, you can clearly see all the layers of retina. This is the inside of the retina. This is the outside of the retina. Inside, there's the posterior chamber of the eyeball filled with vitreous humor. From outside to inside, there are multiple layers of retina. But we at least know that from outside to inside, there are first photoreceptors, then bipolar cells, then ganglionic cells. So these are your photoreceptors, and these are the cell bodies of your photoreceptors. These are the cell bodies of your bipolar cells, and these are the cell bodies of your ganglionic cells. You can look at the rest of the layers right here. So there's your outermost pigment epithelium made up of cuboidal cells. Then you have your layer of rods and cones. These are the nuclei of rods and cones. Then you have your plexiform layer, and then there's your nuclei of bipolar cells. There's your internal plexiform layer, and then you have your nuclei of ganglionic cells, from which arise axons. These are actually the fibers that end up making the optic nerve. These fibers, they join and form your optic nerve in front of the blind spot. So that's it for all the histological slides under your neurosensory system. For PDF or for the slides, you can visit the link in the comment section.